Hi, everyone. Welcome back to Microbiology. We're going to start with Chapter 7 today, and welcome to Unit 2. This chapter is about microbial nutrition, basically what microbes need in order to survive, and not just about what chemically or like food-wise that they need, but also maybe if they need light or if they can deal with oxygen or whatever. Really? Okay. These are the topics that we'll be going over: the sources and uses of essential elements that compose a cell in general, effective environmental factors on microbial growth, whether that's temperature or whatever, various ways nutrients are transported into and out of the cell. We'll be talking about like osmosis and facilitated diffusion and all that. The dynamics of a bacterial population in a closed culture. And like how you would measure how many alive cells you might have in a culture as opposed to dead ones, and name the types of symbiotic relationships between organisms. Starting off with the nutrition, what am I talking about with nutrition? What are nutrients exactly? They are things that are acquired from the environment and used for any of the cellular activities. An essential nutrient is any substance that, whether in elemental or molecular form, must be provided to an organism. Macronutrients required in relatively large quantities. They play a principal role in cell structure and metabolism. And then micronutrients, which are in trace amounts, usually trace elements. They're usually used for enzymes or like cofactors and enzymes. We also have inorganic versus organic nutrients. So inorganic, do you guys remember your chemistry? Organic things are made of a carbon-hydrogen backbone, whereas inorganic things do not have a carbon-hydrogen backbone. That's the idea. A lot of times organisms get their organic stuff from other organisms. Not always, but sometimes. So this is basically like the types of chemicals that an organism is going to need in order to survive. So carbon, oxygen, nitrogen, hydrogen, phosphorus, sulfur, potassium, sodium, calcium, magnesium, chloride, iron, and all these manganese, molybdenum, cobalt, whatever, trace elements, okay? And where they would come from. Originally, these are their inorganic reservoirs. So they have to come from an inorganic place in order to get moved into the organic places. That's the important thing to remember here. So whether it's carbon dioxide in the air, carbonate in rocks or sediments, or it could be from oxygen in the air getting incorporated into things for oxygen, nitrogen in the air getting incorporated into things like usually stuff in the soil is going to do that stuff. And whatever. So however these things are going to incorporate these things all together to help provide sources of these elements to organisms that might eat them even, or for themselves, of course. So this is the chemical composition of E. coli, normal plain old E. coli bacterium would have about 70% water. We can see that's an inorganic compound. It's going to be most of it. But then if our organic compounds, we have quite a lot of carbon and everything like that, carbon and then oxygen. So it's mostly, we see most of that's going to exist through proteins and then RNA more than anything. So only 3% of it is the DNA. So why is there so much RNA? Well, remember, you need to make the copies of your genes to make your proteins. Well, usually if you want to make like the gene that's going to allow your cell to let nutrients in, like a little channel that lets nutrients in or something, you might want to have several copies of that gene made so that your ribosomes can work on making several proteins at the same time. And then ribosomes are made up of RNA, and the things that bring the amino acids over, when we're saying like those 3 bases are going to make up for one amino acid, and that one amino acid is going to be carried on something called a tRNA. And the tRNA is what's going to be attaching that amino acid to the other amino acid and so forth. So that's how that's all going to work. So you need a lot of RNA. Everything needs a carbon source for any of the organic molecules that it might have. Any living thing needs a carbon source of some kind. So we have the heterotrophs. So we're going to go through, these are based on carbon sources. Okay. Heterotrophs get their carbon from other life forms. Heterotrophs get their carbon from other life forms. Most carbon sources exist in a form that is simple enough for absorption directly. Many carbon sources must be digested by the cell in order to be absorbed. But then we have the autotrophs, and the autotrophs, auto means like automatic, troph means eat, like automatic eat. So they make their own carbon source. What does that mean? How do you make carbon sources? Well, they literally take inorganic carbon dioxide and fix it in a way to make their other carbon compounds. So they have the capacity to convert CO2 into carbon compounds, and that I mean organic ones. So convert the inorganic CO2 into organic forms of carbon. And they do not depend on living things at all. Where do things get that nitrogen that they need? We need nitrogen for the nitrogenous bases. Those are used in DNA, RNA, and ATP. We also need nitrogen in our amino acids, as we can see down here. There's nitrogen here. N is for nitrogen. Okay, anytime we see N is for nitrogen. At each of these here, those little crooks, each of these crooks is a carbon. And there's hydrogens there too. So just keep that in mind. Yeah. So primary nitrogen source. What? Indispensable. Okay, yeah, I get what it's trying to say. Okay. Sorry, I misread it at first. The primary nitrogen source for heterotrophs is going to be getting it from other organisms, these. So you'll eat a plant and you'll steal that plant's DNA, RNA, and ATP, as well as other things in there, but that's how you would get your nitrogen. That's what a heterotroph, we're heterotrophs, would do. We rely on other things. So we'll have to degrade it into basic building blocks in order to use it. That's how it has to be done. Yeah. Oxygen, obviously we would get our oxygen by breathing, but you can also get it from the food that you eat, like sugars have oxygen incorporated in with them, carbohydrates. We have fatty acids, that acid portion, which is this portion right here. That portion is the acid portion. So that has oxygens in it as well. I mean, we need it to make our own carbohydrates. We need it to make our own like fatty acids and whatever. And then inorganic salts often have oxygen involved in them. Hydrogen sources, where do we get our hydrogen? Overlapping roles. It has overlapping roles in biochemistry of cells where we can maintain the pH. Sorry, there's hair stuck in my hand. Maintaining the pH of the cell itself, forming hydrogen bonds between molecules as part of like chemistry aspect of it, and serving as a source of free energy and oxidation reduction reactions of respiration. We'll get into that more later. Phosphate sources, where do we get phosphorus from? What do we need phosphorus for? Well, we need it for DNA, RNA, and ATP. It's really important for those. So we need phosphorus for those. Originally it will be derived from phosphoric acid and be found in rocks and organic mineral deposits. So then it'll be incorporated by those autotrophs and they'll convert it into an organic form, and then we can use it that way as heterotrophs because we're lazy and we don't make our own. Sulfur, where do we get that? I mean, the environment, rocks and sediments. We need this for methionine and cysteine, which are amino acids. And that disulfide bridge is important for the stability and the strength of proteins. So when I say here is cysteine, here's that sulfur group, And here is methionine. Here's its sulfur group. And so the sulfurs can form bonds literally across one another. It's either two cysteine molecules that form a covalent bond. They're sharing electrons. So that interaction can happen. So this is an amino acid, by the way. I'm trying to think if I want to say anything else. Growth factor. A growth factor, and this is an important thing to know, is any organic compound, such as an amino acid, a nitrogenous base, or a vitamin that cannot be synthesized by the organism. So you have to have it in your diet. These are things like essential amino acids. You need it in order to build your proteins, but your body can't make it. You don't have the equipment. You don't have the proteins to make it. So you'll have to get it from your environment somehow. Okay, so how microbes feed, the nutritional types. So we have the phototroph. Phototrophs are microbes that photosynthesize. I mean, go figure. Light, they get their energy from light. Chemotrophs. Chemotrophs are microorganisms that gain energy from chemical compounds. You have to be one or the other. All right, so we have our photoautotroph. So they make all of their own energy from scratch, essentially. All of their carbon source will be from scratch. That's what the autotroph part means. Remember, that's what the carbon source part is. Heterotroph gets it from organic stuff, from stuff that usually the autotrophs made. Autotrophs get it from the inorganic sources. So photoautotrophs use inorganic sources for carbon and sunlight for their energy source. So that includes photosynthetic things like plants. They make their stuff from scratch. Chemoautotrophs, they use inorganic things from their environment completely for their energy as well as their carbon sources. So this, you don't see these a lot. These will be the archaea and deep sea bacteria, like methanogens and deep sea. Okay, so that is the photoautotrophs and the chemoautotrophs. Now we're moving on to the heterotrophs. So there will be photoheterotroph and chemoheterotroph. Photo and chemo for each one, okay? Photoheterotroph, they utilize the sunlight as far as their energy source goes, but their carbon source comes from organic compounds that other organisms made. So this includes the purple and green photosynthetic bacteria. Then we have the chemoheterotroph. These are the ones that get their carbon sources from organic sources that other organisms made again, and they get their energy source from other organisms as well. Okay, usually organic is what I'm talking about, organic material. So here they're giving us the examples of like parasites and saprobes, which we don't have to get into, but we are chemoheterotrophs. I don't know why they didn't just put humans. Well, yeah, so we are chemoheterotrophs. Saprobes break down the dead things. Sure, they're chemoheterotrophs. Parasites like protozoa. Yeah, sure. They're chemoheterotrophs. You and I are chemoheterotrophs. Okay, trying to think what I want to say. So photoautotroph. Oh, good. We're going to go back through it. They capture the energy from the light and transform it into a chemical energy that can be used for cell metabolism. And they are usually at the base of the food web. Okay, photoautotrophs are usually the primary producers at the base of the food chain. Chemoautotrophs use chemicals for their energy and chemicals for their carbon source. Lithoautotrophs, they don't need sunlight or organic nutrients at all and rely totally on inorganic materials. Chemoorganic autotroph, okay, they use organic compounds for energy and inorganic compounds as a carbon source. So it's a little bit tripped up there. Methanogens, these are chemoautotrophs that produce methane from hydrogen gas and carbon dioxide. They are only in anaerobic, so non-oxygen environments, in the ocean vents and hot springs. The chemoheterotrophs derive their carbon energy from organic molecules, and the organic molecules are processed through respiration or fermentation, and they'll produce ATP. This includes us. We undergo respiration to make our ATP. It's not talking necessarily about breathing. The breathing is part of it, but that's not necessarily what it's talking about. Okay, so aerobic respiration is what we're going to get into. This is what we do. Okay. Aerobic respiration is the principal energy yielding pathway in animals, protozoa, fungi, and aerobic bacteria. It needs oxygen. That's why it's aero. Okay. Glucose and oxygen are reactants, and that means they're getting put in, and carbon dioxide gets given off as a result. That we make energy in the form of ATP, adenosine triphosphate, that we can ATP, break off that P, ADP is a resulting product, but that breaking off of the P helps move things like generate reactions and stuff like that. All right, that's the idea. All right, so this is complementary to photosynthesis. This is where we're using up the oxygen that photosynthesis makes, and photosynthesis is giving off the oxygen that we need. Yes, that's why. Okay. So I've really thought about it weird. Saprobes. Saprobes are free living. They live all on their own. They don't depend on like living in something else. They're free living. They break down dead organisms. So plant, letter, whatever, dead things. Parasites, they get nutrients from living tissue. So they have usually a host that they live on that they depend on for feeding, and the host is usually usually harmed in some way. Okay. So they live on or in the body and cause some degree of harm to the host. That's what I'm trying to say. They're considered pathogens because they can damage tissues and cause death. We have ectoparasites on the body. Endoparasites live in. Endo, in. It's the same as in. So and then we have intracellular. That's even more in. That's inside the actual cells. That's like the viruses, for example. And we have obligate parasites. You guys remember what obligate means? It means it has to do something, like you were obligated to take out the garbage or whatever. So obligate parasites are unable to live outside of the living host. So that's the case with viruses for sure. So microbes, how do they feed? How do they absorb their nutrients? Nutrients must be taken into the cell itself, and the waste materials have to be transported out of the cell. So there has to be a system for that. So transport has to occur all across that membrane, the outside membrane. Has to get in somehow, has to get out somehow. This brings us to the concept of diffusion. Diffusion is the movement of molecules in a gradient from an area of higher concentration to an area of lower concentration. Think about it this way. It's like if you guys put a whole bunch of bouncy balls outside of my office, all the way up to the ceiling, and then I open my office door, those bouncy balls would diffuse into my office to try to even out like the volume difference of bouncy balls. That's diffusion. So it's higher to lower. It just wants to move that way naturally. It's determined by concentration gradient. So it always goes from high to low to make it equal. It wants to make it equal on both sides. And it also depends on the permeability of whatever. So when I say it depends on the permeability, and I said you guys were going to put the bouncy balls outside my door all the way up to the ceiling, just fill up outside my door. And then it's not permeable to bouncy balls with the door closed, right? They can't get in with the door closed. So it's not permeable, does not allow it to travel across. So it doesn't permit it. So if it doesn't permit travel across that barrier, across the door barrier, that's what I'm referring to as far as sorry, I just realized something. Anyways, yeah, so that's what I'm talking about with diffusion. It's always going to want to go from high to low, but that's only if it can. Otherwise, obviously you can't. So osmosis. So getting into this concept of osmosis. I know you guys have heard of osmosis Jones, but when you hear osmosis in this class, I want you to think of actual proper like osmosis. And osmosis is the movement of water across a selectively permeable membrane. So the membrane is selectively or differentially permeable. It has passageways that allow the passage of water, but not other substances. Now, what do I mean? You have the bouncy balls outside of my office. You decide that the bouncy balls aren't getting into our office with that door closed. We got to get something in there. How are we gonna get it in? Now you guys just grab a fire hose and just start filling up that whole area outside my office. Is the water gonna get in here? Yeah, it is. Under the door, probably through the sides of the door eventually. Like, yeah, water's gonna get in here for sure, but the bouncy balls never will. That's selectively permeable, okay? And that is osmosis. So osmosis in isotonic conditions. The external environment is equal to the cell's internal environment. Okay, so that means, so now we're talking about in a cell. If we were talking about bouncy balls inside and bouncy balls outside, so we finally got the water in here, and maybe now there's bouncy balls too. Okay, but I opened my drawer. I've got 5 bouncy balls in my drawer. So now there's 5 bouncy balls. I'm floating around, not probably alive anymore. And then bouncy balls are floating around. Water's all in, water's all outside. And there's 5 bouncy balls outside. Or at least if you could imagine in the whole setting of this whole office, 5 bouncy balls equally distributed throughout here, that same concentration. So it might not be 5 out there. It's a bigger space out there. So maybe it's like a higher number, but they're all like equally as spaced out as these. That's the same concentration. That's isotonic. Okay. That's isotonic. The external environment, the out there, is equal to what's in here, concentration wise. So diffusion of water. Does it proceed at the same rate? Now, how would we know? Yes. So technically it's always going to be kind of moving around. Water's not going to be steady or anything like that. It kind of always responds to the environment as needed. So diffusion of the water will be the same on both sides. So just basically say it's fine. It's equal. So nothing's really going on, right? There's not really many major exchange there. So that is what we consider the most stable environment for cells, right? It should be happy with that sort of environment. The same inside as out. Hypotonic, solute concentration. So that's the things in the water. We're talking about the bouncy balls. The solute concentration of the outside environment is lower than that of the cell's inside environment. Now, if we wanted to simulate something like that, like obviously the water in here and the door aren't going to respond the same way as a cell, but in general, we have like, I don't know, 40 bouncy balls. I don't know, even maybe like 1,000 bouncy balls in here with the water and none at all out there now. That's what we're talking about, right? So the bouncy balls, they would want to be out there, but they can't be. So how would the water compensate? They can't go through, right? The bouncy balls can't go through. Bouncy balls are stuck in here no matter what. So imagine that we had unlimited space in here and unlimited space out there. What would the water do to try to compensate for that? I'm talking about osmosis. I'm not talking about movement of any other molecule. Let's say there's, I don't know, 3 bouncy balls out there. I don't know, 50 bouncy balls in here. How would the water respond to get that to be the same? We'll only do so much, of course, but like it would move, the water would move from in there to in here to try to make the concentration equal out. But it wouldn't be able to probably because this is so much smaller than out there. So it would be limited by that. The same as with the cell. So what would happen in a cell if that was happening? If the water was moving into the cell because there's too much pure water. There's pure water outside of the cell and saltiness inside the cell, or, there's stuff inside the cell. There has to be, because there's always stuff going on in there. So there's less of less water in there. So the water would want to come in. What happens to the cell when that happens? Well, it will bust open, unless the cell has some sort of thing in place to deal with that, because there's organisms that can swim in perfectly, pure fresh water, right? So their cells have to be able to deal with that. So that's the idea with hypotonic solution. The tonic outside is hypo, it's low. Yes, so this is the summarizing what I was just saying. Pure water is the most hypotonic because there's nothing in it. The net direction of osmosis, that's the movement of water, is from the hypotonic solution, which was outside, into the cell, which is into my office, right? Definitely remember these. Cells without walls can swell and burst because all that movement in here. So I've got walls, so you can try to fill it with water. I mean, eventually you just won't be able to put any more water in here. It's just gonna, that's it. That's all it takes. All right. Hypertonic. So here, the outside has the higher concentration of solutes than inside. So high osmotic pressure will force water to move out. So this is the opposite situation. So if I had, one bouncy ball in here, and there's 20,000 bouncy balls right out there, and we're both filled with water, then the water would want to make it the same concentration. So the water would move all out there, right? To make it the same concentration, to try to spread them out like they were in here. So that's what water would do for osmosis. So the water is moving. That's diffusion of water, right? So the water's still moving by diffusion. It's just water that's being diffused. We can't move the bouncy balls because they don't fit. So this is why you would want to have high salt and high sugar to preserve foods, because things can't grow in that. Like all the water would get sucked out of those cells. You get it? I hope so. I can't get any answers from you. It sucks, but so this is just going over that. We've got isotonics. That's the same on both sides. Everything's happy. Everything's looking pretty good, right? Hypotonic, we've got less on the outside than we do on the inside. So it tries to make it equal. So the net diffusion of water is into the cell. This is going to swell up the cell, make it all big. If we have hypertonic solution, we have a lot on the outside, and on the inside, not really any. And so it's trying to make it equal. So we're just shooting all of our water out. Now, here's introducing this concept of plasmolysis or plasmolysis, whichever one you want to read it as. The water goes out and shrinks it, and the cell membrane is getting, the inner membrane in bacteria, right? It's going to shrink away from that cell wall in bacteria. And that's called plasmolysis. Real quick. Anyways, so you can see clearly that being isotonic is going to be more favorable for a cell for the most part than the hypotonic or the hypertonic. There are cells that are made, like suited for those hypotonic or hypertonic, depending, but they're specialized for that. The average cell, the typical one, that's not the case. All right. So yeah, this is those adaptations. So isotonic conditions, very little stress on the cells. In hypotonic environments, like the pure water environments, bacteria and amoeba live in fresh water. Bacteria will they have cell walls that will prevent them from bursting, so they can take up water, but that's all, they have to live like that. Amoeba can use vacuoles that can just move water out constantly. So they're constantly taking up water, like from inside the cell and moving it out. Of course, that's going to require energy, so they have to have some way to deal with that, more energy to deal with that. Then we have those that are adapted to hypertonic, so they can live in places like high salt and high sugar environments. So the cells must restrict the loss of water to the environment or increase the salinity of the internal environment. So halobacteria, halo being like halogen, so that's like salt ones. Halobacteria like salt, they absorb the salt from around them to make their cells isotonic with the environment, but then they're not hypertonic, then they're isotonic. But yeah, you get the idea, right? So they take up, so they're made that way. They want to be that way. There's also this stuff called facilitated diffusion. That is like a transport that is helped. So we're still moving with regard to diffusion, but we have help for the diffusion. So we have a carrier protein that binds a specific substance, and it binding it triggers the carrier protein, so the substance will move across the membrane. When we look at simple diffusion versus facilitated diffusion, substances in simple diffusion, that's just natural, but we'll move from high gradient to low, like from high to a low with the gradient. In facilitated, we're still moving from the high concentration to the low concentration, same thing. It's just like, in this case, it's like drilling a hole or like bashing a hole through my door so that you could get the bouncy balls in. You get what I'm saying? Like there's a way for it to get across. So in this case, it would be like if I had like a window where like I open, you open one end and close, put the bouncy ball in and close it. And then I open it and I take a bouncy ball, and we just keep going on and on like that until it's equal on both sides. So that's the way that we could deal with the bouncy ball problem. Adaptations. So saturation refers to the rate of transport increasing as the rate of the substance concentration. This is not worded very nicely, I don't feel like, until the binding sites are occupied. So it can only move those things. So if we only have saturation, refers to like if the bouncy ball situation, I'm pointing at my door, I know you can't see it, but the bouncy ball situation, there's only one bouncy ball window. So if we're constantly like got a bouncy ball in there, then that's saturated. That's what saturation is talking about. If we had two bouncy ball windows, but only one needed to be used to get us to equal levels, then that's not saturated and it doesn't need to be saturated. All right, competition. We have two molecules of the same shape that can bind to the same site on the carrier protein. The chemical with a higher binding affinity or the chemical in higher concentration will be transported at a greater rate. So we've got bouncy ball. Let's say that we have another ball that's a little bit bigger than the bouncy ball, or it's like a fluffy ball, like a stuffed animal, okay? A little bit bigger than the bouncy ball hole, but you can shove it in and I can pull it back in, right? You can, but like it's easier and faster for me to do the bouncy balls. So that's going to get transported at a greater rate. So that's dealing with that bottom one. If we're talking about the higher affinity, it's like it has to fit through it exactly. So higher affinity. So it's like if we had it exactly shaped as a bouncy ball, but we're trying to shove the thing in there, it doesn't quite fit, but you can make it fit and whatever. So the bouncy balls are still going to be preferred, right? So they're going to get transported more. That's what that's talking about with competition. Stuffed animals versus bouncy balls. All right, features of active transport. Active transport is transporting anything against the gradient. So y'all got those bouncy balls outside my door and you're trying to shove them through and I don't want any. So I start shoving them back. That's active transport. It's going against the gradient. And it's usually at a faster rate because we're putting energy into it. So that's going to include things that we call permeases. So permease, because it permeates, because there's like literally making holes in the membrane, or pumps that are actually actively pumping things out. This is like the sodium and potassium pumps that you hear about in like physiology and stuff. So these are specialized pumps. They'll carry ions like potassium, sodium, and hydrogen across the membranes. So we can couple the transport of a nutrient to a substance that is immediately useful inside the cell. So that's group translocation. So if you're pushing out like, hydrogen ions and you want to move in like something actually that you want, that can be like help change the protein's shape by pushing those through. There's chemical interactions there, causes that permease or whatever to change shape, and then it can bring something else through instead, or whatever, cause another reaction inside the cell. Endocytosis. Endo, remember that's going to be in. So now we're talking about into the cell, cyto. Transporting large molecules, particles, liquids. So this always requires energy, just like any other. This is another version of active transport. It's just active transport tends to be across the membrane, like in a channel. Typically, then endocytosis is like going to be a bigger movement. So we have phagocytosis, which is like we said, eating. That's phago means eating. So the cell is eating. Pino is drinking. Yes, and you can think of Pinot Grigio if that helps. Cytosis, so cell drinking. So this is just smaller amounts. So oils are molecules in solution. So this is drinking. And the phagocytosis is larger matter. Could be whole cells even. Smaller cells typically, but could be whole cells. Okay, environmental factors that microbes can experience in their environments. Heat, cold, gas, acid radiation, osmotic pressure, hydrostatic pressure, and other microbes. So how do we deal with temperature? So the cardinal temperatures are range of temperatures for a given microbial species. The 3 cardinal temperatures we're concerned with: the minimum it'll live at, the maximum it'll live at, and its optimum. Okay. So we're going to talk about each of these. This is just showing the growth curve, or not really growth, I guess, yeah, I mean growth curves for each of these at their temperatures and what type of organism grows at these temperatures. So the psychrophiles are organisms that have optimum temperature below 15 degrees Celsius. They can grow at 0 degrees and cannot grow above 20. Psychrotrophs grow slowly in the cold, but they have an optimum temperature between 15 to 30, so more like room temperature. But they can grow in the cold, but just slowly. Mesophiles are most of the ones that we would typically be encountering on our day-to-day. Their optimum growth temperature is 20 to 40. Most of the human pathogens are going to like it at 30 to 40. And then we have, so those are the most medically significant ones. Then we have the thermoduric microbes that survive short exposure to high temperatures, but they prefer to be at the mesophile temperatures. Then we have thermophiles that prefer heat. They grow optimally at temperatures above 45 degrees Celsius. They live in soil and water associated with volcanic activity, compost piles, habitats directly exposed to the sun, whatever. Range that they grow at is between 45 to 80 degrees Celsius. So optimal at 45 degrees Celsius. As far as gases, being exposed to different kinds of gases, oxygen has the greatest impact on microbial growth. So that's why most of the time we will refer to organisms as aerobic, anaerobic, or facultative anaerobic. Okay, so microbes can fall into 3 categories: those that use oxygen and can detoxify. So they use it, they use oxygen, and they can detoxify it. What do I mean by that? We're going to get into it, so don't worry. But that's the aerobic. So they use oxygen, they're aerobic. Then there's the ones that can't do either. They can't use oxygen and they can't detoxify it. Those are the anaerobic. It says not aerobic. So they do not use oxygen. No oxygen for those guys. Then we have those that do not use oxygen, but can detoxify it. Okay. Weird. Okay. How microbes process oxygen. Right. So how do we deal with oxygen? If we're a microbe that is exposed to oxygen on a regular basis, how do we protect ourselves from byproducts of oxygen? So oxygen, whenever it is in the environment or whether we're breathing it and we're using it in aerobic respiration, we make these byproducts that can be pretty damaging to the cell. Okay. So oxygen enters into cellular reactions. It's transformed into toxic products. We have singlet oxygen. It's a reactive molecule produced by both living and non-living processes. And then by non-living, I'm not talking about dead things. I'm talking about things that aren't alive, okay? Like just normal chemical reactions in the environment. Buildup can destroy or damage a cell. Then we have these others. We have the superoxide ion and hydrogen peroxide and hydroxyl radicals. Okay, so we have to be able to do it with all four, these three and the one a minute ago. So these ones are disruptive metabolic byproducts of oxygen. So in our normal respiration stuff. Cells can use enzymes to scavenge and neutralize these byproducts, turn them into things that we can actually use. How do we do that? We use these enzymes. Anything ACE is going to be an enzyme. That's a protein that usually typically helps a chemical reaction along. Okay. It's a catalyst. So catalase turns hydrogen peroxide into water and oxygen. Now, what type of a thing is oxygen? Is it a liquid or it's not a trick question? It's a gas, right? Right. We breathe that. So if you are turning hydrogen peroxide into water and oxygen, what would it look like? We just turned a liquid into a liquid and a gas. What would it look like, do you think? It would be bubbly, right? So when you see hydrogen peroxide fizzing, this is the reaction that's going on. Whether you're doing it in your cut or you're going to be doing it in the bacteria in the lab, this is the reaction that's going on, okay? Then we have superoxide dismutase. What does it do? It takes that superoxide ion, superoxide here, and just free protons or whatever in the environment. And then superoxide allows us to convert it into hydrogen peroxide and dioxygen. So just oxygen, like the oxygen gas, it's dioxygen, okay? So what do we do? So, oh, great, you made that hydrogen peroxide. Thanks, Dad. Like, no, if you have superoxide dismutase and you're making hydrogen peroxide, you're going to be able to have catalase deal with it after that, right? So they'll work together. All right. Okay, so aerobes. Aerobes can use gaseous oxygen in its metabolism. So we talked about this. Aero means literally air, meaning oxygen. It's okay to live out in the air. Okay. They possess enzymes that they need, those ones we just talked about. If you deal with air, then you have to have something like that to protect yourself. Okay, so they have enzymes to deal with those toxic byproducts. Obligate aerobes, they're obligated. So if you're obligated to take the trash out, you have to take the trash out, Mom said. So obligate aerobes have to have oxygen. They cannot grow without oxygen. Obligate aerobes cannot grow without oxygen. Then we have facultative anaerobes. So facultative, what are we talking about here? Just think of facultative as they could go either way. They could facilitate either way. And these are aerobes, essentially. So they can use air. I don't know why it's anaerobes. It does not matter if it's anaerobe or aerobe really for these. Okay, just let's pay attention to the facultative part. And I was tricking you with the second part there, I swear. Facultative. If it's facultative, it's an aerobe that does not require oxygen for metabolism. Doesn't need it. Metabolizes using aerobic respiration when it does have oxygen. When there is oxygen, it will do aerobic respiration. What's aerobic respiration? It means it uses oxygen, right? That's aero. Anytime you see it, oxygen, okay? So anytime there's oxygen present, it will do aerobic respiration. When there isn't oxygen, it will do anaerobic metabolism. In this case, fermentation. Okay, microaerophiles. They require a very, very small amount of oxygen in the metabolism. An example of a microaerophile is Campylobacter. You guys have ever heard of Campylobacter? It is a pretty common bacterial diarrheal disease, like foodborne illness. Not something that you necessarily call like the CDC over or whatever, but like people just get it all the time, like during like Thanksgiving or whatever, because they don't cook their turkeys enough or whatever. Very common. Microaerophiles, very, very, very micro was little, right? We're learning that in this class, little tinies. And then aero is oxygen. All right. So anaerobes, here we go. Anaerobes, the ones that cannot aero, they lack enzymes for using oxygen. So they would die in the presence of oxygen. Strict or obligate anaerobes cannot tolerate oxygen. Aerotolerant. There are some that are aerotolerant. So oxygen tolerant ones that don't use oxygen. All right, so that's what that wording is going to mean. Aerotolerant anaerobes. They do not use the oxygen, but they can survive it to a limited extent. Then we have capnophiles. These are at high CO2 than what is normal in the atmosphere. So we typically would grow them in incubators that we can provide a controlled amount of carbon dioxide for. I mean, I don't know how else to make this one except, I mean, capno CO2. I don't know how to make that cute. pH, regarding how they can grow in the presence of certain pHs. Obligate acidophiles. These are things that are obligated, so they have to do it. They have to take out the trash, mom. Acidophiles, right? So here we're talking about acid. They require it. Obligated. They have to, so they will require acid. Okay. This includes molds and yeasts. This is not uncommon to think about if we were to talk about some of this stuff in terms of like, you know, foodstuffs and everything. That shouldn't be too surprising or weird, right? When you really put it into a real life scenario, this stuff isn't that weird. So alkalinophiles. This is alkaline. Do you guys remember that what that word means? It means high pH, right? Basic. So they like it to be high pH. So that's hot pools, soils with high levels of basic minerals, things that decompose urine and create alkaline conditions. I mean, that makes sense, I feel like. Osmophiles, so osmotic pressure now. Osmophiles, they live in habitats with high solute concentration. They love the water moving across. So the high solute concentration. So this is things like the obligate halophiles. Obligate because they are required. Halo was like halogens, like salt. High concentrations of salt. Yeah, so they have high significant modifications to their cell walls and membranes and will lyse in hypotonic habitats. They will die for sure. Facultative, so they can go either way, right? Facultative halophiles, salt. They can resist salt. They can deal with it. I don't know if that's how I would say. They can deal with it. In the presence of radiation, phototrophs have to be able to deal with this, right? Because they're in the sun, that's like their whole point. So phototrophs can use visible light rays as an energy source. So they need protection. So how do they protect? They have special pigments to help protect them. It absorbs the energy of the radiation itself, or they have special enzymes to repair UV damage. So as the ultraviolet rays start causing problems between those base pairs in that ladder, in the rungs of the ladder, instead of the base pairs pairing this way, sometimes some of them will pair like next to the one next to it. And that's what happens with UV damage. And then that causes problems in DNA replication and stuff like that. So they had to have the ability to deal with that. Hydrostatic pressure, these are the barophiles. They can deal with deep, deep sea, so high, high pressure on them. If you bring them back up here to the normal pressure, that they will rupture. This happens to organisms down there too, if they try to bring them up. Okay, these organisms, they interact with one another. They live with one another. They have lives together. They live in similar environments. How would we classify these, interconnections? Some of these are important to understand. So we have symbiotic versus non-symbiotic relations, right? So just so we know, symbiotic means these organisms live in close nutritional relationships. One or both members in the relationship will require it. They actually require it. In non-symbiotic relationships, the organisms are free living. It's not required for any of them, but it can benefit still. So we'll start with the non-symbiotic ones. We have synergism, where members will cooperate and share their nutrients, versus antagonism, where members are inhibited or destroyed by others, depending on like what their activities might be in that ecosystem. These are not as important. The ones I want you to focus on are the terms here for we do want you to know those, synergism and antagonism, but definitely focus on the symbiotic, mutualism, commensalism, parasitism. These are going to come up all the time. So know these and they'll be on the test for sure. Mutualism. Both members are going to benefit. They both depend on it. They both benefit. Think of your gut microbes. Your gut microbes are getting protection and getting food from the food that you eat. You're benefiting because they're giving you protection from like outside invaders, like, food poisoning or whatever, as well as like they call it microbial antagonism. They break down some of the foods that you can use their, like the byproduct breakdown as nutrients for yourself. So that's mutualism. We're both benefiting from that. Okay. In commensal, the commensal organism is the one that benefits. The commensal benefits. The other one isn't harmed or benefiting. It's just there. Parasitism. It's pretty self-explanatory. If you had a parasite, it sounds bad. It is. Parasite. Parasite depends on the host and it's benefiting. The host is always harmed in this relationship. Okay. So going through these a little bit more in detail, symbiosis, two organisms live together in close partnership. That's all symbiosis means. Your lives are intertwined somehow. Somebody requires being there though. Mutualism means you are obligatory, but mutually beneficial relationship. Obligatory, like we said, obligated, right? Obligate, like obligate anaerobes, obligatory. They have to happen, has to be that way. It's required. Commensalism. The commensal is the organism that receives the benefits. And I say the other one is not harmed or benefited. The word for that other one is co-inhabitant. Okay. Parasitism. Host gives nutrients and habitat. Parasite harms the host and gets the nutrients and habitat from the host. Satellitism is just one member provides nutritional or protective factors needed by the other. Don't worry about that one too much. Antagonism, when members of the community compete, one microbe secretes substances into an environment that inhibits or destroys other microbes. So this would be the case a lot with our gut. Like if something else, we did eat some sort of food that had bacteria in it and it made it that far. And there are microbes in our gut that can produce toxins that are fine for us, but they're toxic for incoming invaders. That would be an example of antagonism. Antibiosis is a form of antagonism where we have the production of inhibitory compounds like antibiotics, like the mold does. So if you grow mold on a plate, it might have the production of natural antibiotics to prevent the growth of bacteria into its habitat. Antibiosis. Synergism, an interrelationship between two or more free living organisms that benefit, but is not necessary. So this is the similar thing to mutualism, but this is not obligate. This is not obligatory, right? This is not necessary. All right. This is the perfect example of synergism, the epitome of synergy. Biofilms. Biofilms are mixed communities of different kinds of bacteria and other microbes. The pioneer bacteria attached to the surface, they're living, they're growing and multiplying. Other microbes also attach because they see that stuff is going pretty good for them. And they have this ability, this is an important one for you guys, quorum sensing. A quorum is like a group of people where you like come together and you talk. It's almost like a Congress. And you would talk about like whatever's going on to make sure everybody is aware of the situation, like a town hall meeting or something. So they have quorum sensing. And the cells in the area are stimulated to release chemicals, it could be chemicals that are actually releasing for the situation, or even just the byproducts that are making from whatever they're eating in that area. Whatever it is, the organisms in the whole biofilm are constantly monitoring it to make sure that everybody in the biofilm is growing well and feeling well and doing well and whatever, to monitor the whole size as well, so that they know where they stand in all of this, or if they're about to get like lose all of their food source because there's so many of them or whatever. So this is that. Pioneer bacterium come in. They secrete extracellular material that helps them adhere, and other organisms that come to join them to stick as well, right? They start quorum sensing. Quorum sensing. The chemicals are the red dots. All right, in many biofilms, other species join and contribute to the extracellular matrix and or participate with quorum sensing with their own chemicals or the ones released by the other species. Could be the same chemicals they're all using. Either way, whatever they're making, their byproducts or chemicals or whatever, allows everybody to know kind of what's going on with the biofilm. So mature biofilms may stop or minimize quorum sensing. And biofilms serve as a constant source of bacteria that can escape and become planktonic. Again, literally plankton is what we're talking about there, but free living. They can just exist like that. Okay, we have other relationships with microbes, right? We have a normal microbiota. We're going to talk about this so much in Unit 4 and Unit 3. Unit 3 probably more. Unit 3 probably the most, but definitely in Unit 4 too. Yeah, both of them. Sorry. Normal microbiota, microbes that live on the skin, in the alimentary canal, or other sites or whatever. They could be commensal, parasitic, synergistic, mutualistic with the human host. Okay, the doesn't always have to be good, but that's our normal microbiota. Stuff that's always there. All right, growing bacteria, how are they going to grow? They grow by this thing called binary fission. The parent cell gets bigger, the chromosome duplicates, the cell pulls apart in the center to form a septum division, and then cells will divide into daughter cells. That's pretty straightforward. But yeah, young cell makes chromosome double, gets bigger, makes a band in the middle of the septum that helps it squeeze it in the middle and then make two daughter cells. That's binary fission. So one becomes two. So that's going to be what we use to determine the generation time or the doubling time of a population. It's the time required for a complete fission cycle for one to become two. Each new fission cycle doubles the population, right? So 2 becomes 4. 4 becomes 8, and so on, like it says here. As long as the environment remains favorable, it'll keep doing that at a constant rate. The length of time is a measure of the growth rate of an organism. So then we can get into talking about exponential growth. Essentially, that's when it starts growing like, the numbers get higher and higher and higher, and the curve is actually more of a curve, like an exponential growth curve. It's a logarithmic curve. So growth pattern of microbes used to express population microbes as exponents or logarithms. I would say logarithms is probably the most commonly used. We use log 2 or natural log, log base 2, not log base 10. Normal, that's pH is log base 10. So, so this is exponential growth though, still. Plotting the data. Yeah, we'll plot them as a function of time. I mean, that's pretty straightforward, right? How many are existing at each time. So this is great, but like, how do we actually get those numbers, I guess? So a growth curve that we were just looking at is a predictable pattern of growth in population. We want a viable count technique though. If we want to know how many of them are growing, we need to know how many of those that we're counting are alive, not just like we look under a microscope to count cells and then we can't tell which ones are alive or dead, right? What if they're all dead? How would you know? So that's what we want, is a viable count technique. Count cells over a given period of time. That's live cells. So there's how it might work. You have a liquid medium here. You add, I don't know, 1 ml or 0.1 ml, depends on how much you have in here, right? But you can dilute it out like 1 to 10 or 1 to 100, as long as you know what your dilution is. And then you plate it over time. And then, or you would check it, like there's checking it here over time. So this one's after 600 minutes. So they've got so much growth after that amount of time. They've diluted it down and they count out the colonies of how much is growing. Now, they can't grow colonies if they're dead. So now you know approximately what your growth curve is going to look like if it's alive. This is how you might track growth of something in a culture like that. It's important to know all these phases. There are 4 phases in a growth curve. Okay, this is a normal growth curve, 4 phases. You have the lag phase, the exponential growth phase, the stationary phase, and the death phase. And I'm going to talk to you about what each of these is. The lag phase is that flat period on the graph when the population is just not really, it doesn't seem like it's growing, right? What's going on there is they are newly inoculated cells. They need to adjust, they need to enlarge, they need to synthesize the DNA before they can start dividing and growing and everything. So they're just not yet growing at their maximum rate. So, yeah. So then next we have the exponential growth or log phase. So go back to here, we did the lag phase. They're just getting ready. They haven't yet grown or started dividing yet. The exponential growth phase is next. I mean, the growth curve is going to increase dramatically. Everything's doubling. Phase will continue as long as cells have adequate nutrients and the environment's favorable, no toxins or anything. Remember, some of the toxins, some of the times whenever they get to the death phase, they produce toxins that are not friendly for growth either. So stationary growth phase, that's where it's flat next. So let's go back. That's that third one here. Why is it flat like that? The cells are in survival mode. They're just trying to kind of exist. They stop growing or they grow slowly in the stationary phase. And that means you're coming up to the death phase soon. So limiting factors intensify. So like there's not as much nutrients available, or there might be toxins now, and the cells will die at an exponential rate, exponential just like they grew. Okay, so the curve will dip downward. So antimicrobial agents can rapidly accelerate the death phase, obviously, because they're going to create more of those limiting growth factor situations. So microbes in the exponential phase are actually more vulnerable to those agents because they're actively dividing. So you can inhibit at that point, because if you add in something that's going to stop them from growing, typically a lot of them are going to affect them when they're growing, like stop them from producing proteins or nutrients, or affect their cell walls or something. So it's more useful when they're actively growing. They're more vulnerable that way. Otherwise, they could just be sitting there doing nothing. So then you can disrupt their cell metabolism and their binary fission steps by adding it during that growth phase, the exponential growth phase. So growth patterns can account for stages of infection as well, depending on the growth rate of whatever microbe we're talking about. The growth curve, the chemostat. So this is a continuous cell culture system to siphon off media and old cells to keep the culture just happy and active, so it never dies. If you need a culture to study some bacteria or whatever, that is what will be used for that. So other ways you might analyze population growth. Turbidometry. You would just be measuring, geez, it is loud. Be measuring how cloudy it is. The greater the turbidity, the larger the population, right? So the more cloudy it is, the more bacteria are in there. You can even dilute it out if you're not getting a good read, if it's just too cloudy for it to read. Then you can do the direct or total cell count. You can count cells in a sample that, like using a microscope. We'll use a cytometer to analyze a tiny sample that's spread over a pre-measured grid. I'll show you a picture of that here in a second. But then that's the question, right? How do you know if you're counting live or dead cells? So you would want to do colony counting after dilution, like what we were showing previously. It does take time. That's the problem, right? Is there any other ways that could be quicker? There are. But first, this is that this is that culture counter. This is that counter that we would use to count the bacteria. There's like squares on it, and then based on what square it is, you count how many bacteria are in the square, and that would determine, you multiply up to determine how many bacteria are in your culture under a microscope. So culture counters are this is the, let me show, oh, you can't see it. So each cell is analyzed by an electronic sensor. It's literally like an electronic sensor, and it's just dripping it through that sensor, and it's counting like how many go through. So do you think this is going to analyze alive or dead cells? Yeah, you can't tell. You can't tell with this one. So now we have a flow cytometer, and this is similar, right? So now we have a laser that we're going to shoot. And we can do 1 by 1 cells, and this can measure the cell size, and it can differentiate between live and dead cells based on how they stain. So we'll use fluorescent dyes for this. And so yeah, so on this one, you can tell that whether it's live or not. So this is the idea with that culture counter. And similar concept with the flow cytometer. It's just like it can analyze different colors and all of this stuff. So other methods to analyze population growth, we have polymerase chain reaction to quantify microbes in samples without isolating and culturing them. What is polymerase chain reaction? Do not worry, we're going to get in detail, but not right now. Polymerase chain reaction, PCR, that is using a little bit of DNA, and you can amplify it up just to, ridiculous amounts with just a small sample to start with, and amplify it out and analyze it. So like you can even do it like based on, you can use special primers like we'll get into the details of it later, but to identify whether it is salmonella, and special ones to identify if it is E. coli, and then you'll have an answer in like an hour and a half is if it's E. coli and what strain of E. coli it might be and stuff like that. So it's super useful and very, very quick. You don't have to wait days for things to grow and all that. So this is a method of, PCR is a method of amplifying DNA to identify specific microbes. That's a very important thing to understand. And also can quantify, yeah, as well. Just because, depends on how quickly it's going to grow up the DNA, right? So it's more in there, then it's going to be faster. So tests that measure ATP can also be used. This is literally just going to be determining, breaking cells up and seeing how much ATP is in there, and that should be used to, if there's a lot of it, and then there's a lot of cells growing in that culture, right? So you get a little sample of it, not the whole culture, of course. Right, so that's rapid quantification of microbes. It gives a rough estimate of how many microbes are in there, and it's not perfect. And you can also test for accumulation or depletion of chemicals that are in the growth medium. So summarize, we talked about the essential nutrients. So these are the big ones to be aware of: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. That is important to understand that. Chomps. Oh gosh, it's good stuff. All right. Then we went through carbon sources as well as energy sources. Those are different things. You need carbon in order to make things that you're made of, but you need energy sources to use to help you build those things up, right? We talked about transport of nutrients. We talked about diffusion versus osmosis, diffusion in water, endocytosis, endo taking in, and cyto cell. Environmental factors involved in growth of bacteria, temperature preferences, osmotic pressure preferences, like hypertonic solutions and stuff. And we talked about symbioses. We talked about how things live together in biofilms. And then we talked about bacteria growing up. So that is it for chapter number 7. And then chapter number 8 is going to be microbial metabolism, and it is going to be rough. So get prepared for that one. We're going to talk about aerobic respiration and things like that. So brush up a little bit, go back to that chemistry chapter, chapter 2, and just refresh your memory a little bit on some of that pH and everything like that, because we are going to be using some of that stuff and talking about that stuff in that chapter. So I will see you there at chapter 8 next time.